the higher area. So each one here, we've done imaging in specific transgenic animals in a specific layer of a specific set of neurons. Each time roughly 300 neurons in layer 2, 3, on layer 4, on layer 5. Always under the same condition, always seeing the same type of stimuli. So you can now, and people begin to do that, you go again to the website, download all this data, and then you can do lots of analysis with the statistics and how and correlation, function, connectivity, etc., etc. So right now there are 30,000 neurons there, but every 3 months we're adding more under very standard conditions. Then you can also do what you can do in a mouse is 1P whole brain imaging, where you can see this is not averaged. This is individual trace. You have a mouse running, he can see its speed, and he can see its, if you want, it's hemodynamic, it's one photon activity in an EMX mice, and here you can see these specific 6 regions. Lots of interesting stuff going on. And then the third tool we're using, so we developed over the last 5 years in a consortium under the leadership of Tim Harris at Genelia, together with UCL. We went to a foundry called IMEC in Belgium, the big circuit foundry. We built these probes. They're fantastic probes. I'm not going to go into the detail. So essentially, there are 966 probes here, spaced 20 microns apart, and you can select in real time 384 of them. So it's 10 pieces. It's 10 millimeter long, sorry, like this, thin, thinner than a hair, 20 micrometer, thin, 20 by 70, minimal damage. And so it's very dense. That's the most important thing. It's very dense. The best thing is the data at the base of the electrode is digitized, amplified, digitized, and multiplexed. So you don't have 384 wires going out, because that was one of the limits of the current existing technology. You record from 36 neurons, and you have like electrodes, and you have 50 wires going out, you get the spaghetti. You only record from 12. So this it's going to be a game changer, this technology. So for instance, in a small animal like a mouse or rat, you can record pretty much along the entire track here, the neurons everywhere. They're being tested now in primates, and in principle they can also be put in, for example, in human surgery in the human brain. And you just have this beautiful data where you can see, this is only for experts, when you record and you can see activity in visual cortex, in the hippocampus, in dental gyrus, in the underlying LGN, all along a single track. And the beauty of it, spike sorting that used to be a big challenge in the field is becoming very easy. Why? Because each neuron now is imaged, if you want, electrically speaking, on average 9 different electrodes, because the electrodes are so close to each other. These are low impedance electrodes. They're so close to each other that the spiking of one electrode, essentially electrically speaking, you can see on 9 different electrodes on average. That makes the spike sorting much, much easier. So here we get yield per structure, typically between 50 and 90 on these different structures in 2 different labs. It's done both us at Seattle and in London to show the technology is very robust. The yield is very high. It's really this is going to be a revolution. They'll be publicly available next year for roughly €1,000. Then we're doing functional connectomics. We now have 5 of these high-density EM arrays. So this is a project that 2 PIs, I'll just talk one. One is Stephen Smith, who joined us from Stanford. The other one is Clay Reed, who joined us from Harvard. So he, together with Nuno, this is part of a very large IARPA project called Micron, where the idea is, in order to improve deep machine learning algorithm, we want to understand how biological systems do learning. And so here the first goal is this is a collaboration with Andreas Toliers in Texas at Baylor and Sebastian Sang at Princeton to do imaging in a mouse brain of a cubic millimeter. That's the goal ultimately. A cubic millimeter of imaging where you functionally identified the functional performance, for example, the orientation, direction, selectivity of each neuron in this cubic millimeter in visual cortex. You then cut it into 25,000 slices, each of which is 35 nanometers. And so now we're talking 3 orders of magnitude lower, because this is electron. This is not imaging with photons, with electron. Where the individual pixel is 4 nanometers, and the things are spaced 35 nanometers apart, and then you try to move towards automatic reconstruction and automatic annotation of all this data using machine learning tools. So as I said, this is done with Sebastian at Princeton and with the team, with the Neuromancer team at Google. It's really been a game changer over the last 2 years there, the technology. So in the first phase that we just successfully finished this week, the plan was to take 100 micrometer, 100 by 100 by 100, and to show that you can reconstruct this. So this volume, 100 by 100 by 100 micrometer, includes roughly 1 million synapses. So we've actually done this now. This week we successfully cut into the serial ultrasound section 25,000 sections without leaving a single one and putting them on tape. That's pretty freaking awesome, I might say so. Now that's of course only 1/1000th of the volume that IAPA wants to do in the next phase. So this is 100 micrometer, what we just did, which by the way has never been done before. So technology is really dramatically accelerating here. In the next phase is to do something 10 times bigger, 1,000 times bigger. So to do a cubic millimeter, cutting it, and then reconstructing it, which will include 100,000 neurons and 1 billion synapses. And then you collect statistics on the connectivity, who talks to who, you know, particular with cell type specific information. And so with these, I mean, this technology is just dazzling here. This is the automatic clustering software where you can now do pretty much unsupervised segmentation of spiny neurons. So this is a neuron that was reconstructed at the ground, what we think of world level truth, electron microscopic truth, you know, with pixels that are 3 to 4 nanometers. Right, you can see all these individual spines. Here you can see a whole volume that's supposed to rotate. No, it doesn't rotate, it's supposed to rotate. So I think that's a game changer for us, I mean for the field. Now lastly, we also have a team of roughly 30 people, an analytics team, you would call it from industry, where we're trying to do the following. We're trying to sort of to achieve this virtual loop. So we have very detailed description of individual neurons, those are the nodes. We have quite good description about connectivity, and of course we're getting this ground truth connectivity in a cubic millimeter, where we know every single connection bar none, or 95% of them. Then we have 3 different we have the behavior of the animal, and we have 3 different functional measures. The EFIS, as I said, that goes in very high density across a column. The OFIS, optical physiology, that goes at 30 Hertz among genetically identified population, horizontal population, and then the whole brain, mesoscopic imaging. To put that, use a variety of different models, where we have either point models, GLIF models, of a family of them, of various degrees of complexity, from simple integrated phi to much more sophisticated GLIF. Well, we have biophysical models, you know, ACID models where you have currents in the dendrites and active dendrites and all of that, to quite abstract population statistics and machine intelligent networks, you know, deep networks, deep convolution networks. So to finish off, I just want to show you one example. So we've just done this, it's almost ready to be released using SDK, where we've done our first attempt ever to model in great detail using primarily our own data, the behavior of cells in the input layer of primary visual cortex. So this is very little ambitious. This is just the input layer into primary visual cortex. So 10,000 biophysical neurons surrounded for boundary condition by 35,000 simplified neurons. We know, of course, their shape. We know a lot about their synaptic connectivity, all from our own data. We know a lot about the input, again, because we record from the input, so we can model that quite accurately. We have, so we use different instantiation of sort of different model mice. They all have the same statistics learning rule, but each one is a specific instantiation, and then we can train it against specific instantiation of individual mice of our data. That too. Okay, thank you. We're almost done. So Thank you. So what we can do, we can in great detail model. Now this is quantitative modeling. We can in great detail. I'm not going to bother you with the detail. We can always compare. This is simulation. This is experiment. We can look at a whole variety of things, canonical circuit, amplification, Rodney-Douglas, and things like that. We can compare against optogenetics. And what's very satisfactory that we can reproduce quantitative features, including time constants, how much current amplification is there in cortex. The experiment says 3.5, and we find 3.6. So it's really pretty satisfactory. That tells us why is this important. So I love biophysical modeling because biophysical modeling has to obey the laws of physics, right? The conservation laws, for example, charge and current, etc., have to be conserved. It's very easy with the very abstract models to fool yourself that you're doing something correct. Here you can have the physical data, you're measuring currents and voltages, and you can compare it against that. And so either they match or they match within some certain, some percentage of your metric, or they don't match. And so it really means we're beginning to understand in detail at least these very simple input structures into visual cortex of a creature like the mouse, with some very interesting new discoveries that we are testing, things that we have learned about log normal distributions, about particular types of weights, like-to-like weights that have to be there in order to reproduce the data. So the modeling, so I know from my own experience that the modeling I used to do at Caltech for many, many years was sort of more qualitative feature, because we were so far away from understanding even simple biological things like neurons that just to get roughly, to get action potential, to get dendritic spike was a major achievement. But now, at least our understanding at the level of individual neurons and how they interact at the level of individual elements is beginning to be better and better. And we can put that together in realistic physical models in order to reproduce that. And of course, you all know Feynman's famous quote, what I cannot simulate, I can't understand. So, and we're now going to much larger models. And we'll probably have some sort of design competition where we'll put this data out and then challenge other groups in the field to see who can do better, because we're probably not the best one at this, where we model the entire visual cortex, all 6 layers, given all the interneurons and all the pyramidal cells that we have. And we're also very interested in things like local fields and currents, because we can measure these and we can also measure them in humans. In this work, we're very interested in carrying out moving to human, because in human we do have data, we have very little data about single neurons, but we have quite a bit of data from ECOG and from other devices like intracranial electrodes that surgeons have to put in. And so we can compare, if we upgrade those models to human, from a mouse to human brain, we can then compare it against clinical meaningful measurement and close the gap, the very large gap that exists in understanding the human brain. All right, so to finish, so we are sort of, I've shown you roughly what our team of 300 people can do. There are some unique challenges I just wanted in this very last slide to mention. So we're not like a university, we are with these large teams, and so it requires very different motivation. How do we motivate, very bright, highly inspired people? How do we motivate them to join a large group endeavor, where very often they can't do what they want, but we have to do sort of overall what the group dictates. And if everybody goes off and does their own thing, the group as a whole will suffer. Right? You can run faster by yourself, but you can run farther in a group, and so that brings some unique challenges with it. And with that, yeah, you can also think of it as an experiment in the sociology of neuroscience. And with that, I thank you for your attention.
This is okay. Thank you, Christof. Do we have any question? Okay. Let me turn the light.
Thank you. It's very inspiring lecture, and many thanks for your coming. So, almost somewhat implicitly, but you started your lecture that neuroscience is strong at the microscopic level, at the macroscopic level, and you started, it was mostly about the mesoscopic level. So speaking about cortex, finally, almost at the very last slide, you showed once the term column. But this mesoscopic concept of cortical motor were with us for decades, this is not a neuroscience community, so we don't necessarily know. It was identified in the somatosensory cortex in the late 1950s by Montcastle, and then the visual cortex, and then there was the anatomical demonstration note of Jürgen Tübinger, Reutenberg also was involved. I have Hungarian accent, so Szent Ágottai. So the hope was that basically the mouse and human has the same structure in terms of columns. They contain 5,000 to 7,000 neurons, and the main difference is in the number of modules and the interconnectivity of these modules. And the hope, obviously, is the computational community. It would be good to see there are modules which are structural, functional, and computational units, and probably they are not. So I wanted to tell you, you didn't mention this concept, so you don't need it. But finally, you showed once column, so I don't know how it is. So do we
We don't know.
And yeah, that's a good answer.
We don't know. As you said, it's a very powerful concept column that has been around. The column is sort of the basic processing unit that's unreplicated. I would dearly love to show that it's there, but that requires much more work to show that it's actually very similar in V1, in ALM, in the neighboring region. So we still don't know to what extent it generalizes.
We don't know, and doesn't mean that they don't exist, right? So that
correct. We simply don't know. Right now it's a belief of many.
Okay, first, a wonderful talk. It's the first time that I hear all these details about the Allen Institute, and it was wonderful, like really amazing talk. Thank you so much. So you have all these many kinds of neurons in a very small part of the brain. Do you have all of them or subparts of them in different areas?
We hope so. We don't know. People do believe that there are cell types like layer 5 piranha cells that are very similar in A1 and B1 and S1. But if you look at the genes, they seem to differ, partly because they project to different areas. So this is now, the next 5 years, we and other groups will answer that. Is there a common scheme? Is there something like a periodic table? Is there a systematic gradient, for example, across cortex? If I flatten the cortex out, is there a systematic gradient in terms of the layer 5 cells going from one end to the other? Many people believe so, many anatomists believe so, but we'd like to see is that true in the data.
One more question. The observatory sounds like fascinating. So can you see any changes because of the observatory, like changes in brain, changes in activities?
What we're thinking about now doing, you might like this, doing sort of a learning experiment, right? So what happens if we get the statistics of this before, and then we teach the animal. Then we teach the animal something, and then we see whether the statistics change. That seems like a pretty cool experiment.
So, yes, thanks again for coming here. This is sort of a pedestrian question, but I'm just fascinated by it. So, two parts about the surgery. So, one, when you're doing surgery on mice, do you need microrobotic surgery or anything like that? And then two, who's doing that surgery? Neurosurgeons are expensive, so anyway, those are my questions.
Yeah, so we do, in fact, there's a paper out, we did develop a robot to assist us because we always want to operate at exactly the same position using the same drill burr holes. So we do have a lot of machines that help, but it can't be done fully automated because there's this very sensitive thing that right now we've not been able to do using a machine. No, these are trained because it's always the same thing. They're trained RAs, and they do this fantastic with very high reliability because they're doing this each day 4 hours. We limit them because otherwise they get carpal tunnel. Rather than give the postdoc or give the grad student that does it once and then doesn't do it again for another 4 months, and so you get vastly more variability, and we're trying to engineer all that variability to the extent we can out of the system.
That was, sorry.
Yeah, please.
Okay. That was a fantastic talk, and I think this is a fundamental of the computational evolution. I really do think you're doing fundamental, just so important work. A couple of questions on the neurosurgery that you're doing, that the neurosurgeons are doing on the human. When you get when you get the tissue, how do you know that it's not, you don't know that it's contaminated from disease or anything like that?
It's an excellent question. So here you have to remember, the only time we get a brain is when it's dead, a human brain, right? The only other time you get it is when it's cancerous or when it's epileptic seizures. So everything we do, we have to track that. We track that. We do lots of cytology tests to make sure there isn't any obvious invasion of cancer into our tissue, but it's always in the background of our mind. But that's, that's the nature of human tissue. You're not going to get a young, healthy brain tissue.
But you can have mine. It's not young and healthy, but it's free. The second question I had, you indicated on your electrodes that you had 9 electrodes. How do you account for the field effects between those electrodes? Is it because of the low impedance that you have?
Yeah, but we do have to be, remember they're very, very thin. The shaft is 20 micron by 70 micron. So even a cubic millimeter, but we have to place them, and this again we do using robots, we have to place them so they don't directly get close to each other. But they fall relatively, within 50 or 70 micrometer, there are no more interactions between them. So we can get pretty close.
Excellent, thank you.
Thank you for the very nice presentation. I'm curious about the architecture. We have this vision of the high-level architecture of the brain, thanks to several works in the past, in the microarchitecture of the column, although it's still under study. But do you have a middle ground architecture of the vision system of the mouse already? Because this would help a lot us that study neural networks on how to better model.
Yeah, so there seems to be a big difference between primates, human and monkey on one hand, and rodents on the other. Rodents and networks are flatter, so they connect to more. So they connect more widely in a flatter hierarchy. In monkey, and in humans looks like it that way. We have this deep hierarchy where you have 12 or 14 levels, probably more and more abstract representation. So you can, you have a very abstract representation for Donald Trump, for instance, right? The mouse, the mouse, there's no evidence that the mouse has this very abstract representation, and hand in hand with that goes that the fact that just in terms of anatomy, it's much flatter.
So do you have this data available? Can you go there and see this architecture already?
Yeah, some of that's already published.